Okay, we're going to start chapter 4, part 2 notes. And part 2 will be given in 2 sessions. So this is session 1. All right, so part 2 is going to be talking about the different types of membrane transportation, the different ways that certain molecules get in and out of the cell, and the mechanisms that are used for those different types of molecules. So plasma membranes, cellular membranes, they are selectively permeable. And what that means is that some substances are allowed to go through the membrane and others are not. So they get blocked. Okay. Just to kind of review some vocabulary, if something is permeable, it's allowed to go through. If something is impermeable, it's not allowed to go through. And then obviously selective permeables where some things are allowed to go through and others are not. It's kind of the middle. So all cellular membranes all plasma membranes are selectively permeable. They're going to allow some substances to go through and other substances to not go through. So with our plasma membranes, you have to think about their composition. And so if you think about it, you know that the lipid bilayer has the hydrophilic heads of the phospholipid. Then the middle part of the membrane with the hydrophobic tails, that's nonpolar. And then you have your hydrophilic head on the other side. And so the middle of your membrane, the middle of your plasma membrane is nonpolar. And so what that means is that nonpolar molecules, so these are molecules that are soluble with lipids, so they are the same composition, they're able to go right through the phospholipid bilayer. It's kind of like if a nonpolar molecule comes up to the lipid bilayer, it's allowed to go through because it matches the consistency of the interior part of the membrane. So because it matches, it's allowed to go through. They're both the same polarity. They're both nonpolar. It's kind of like when you talk about substances that dissolve something, you always say like dissolves like. They're the same kinds of things. And that's the same idea with our plasma membranes. If you have a molecule that's trying to get across, if it's the same makeup as the middle of the membrane, then it can go right through. If it's not, it can't go through. Okay, it doesn't match, so it's not able to go through. So all nonpolar molecules, they're able to go right through the phospholipid bilayer with no problem because they're of the same consistency as the middle of the bilayer. Now, water is also able to readily go through the membrane. However, as you know, water is a polar molecule, and the internal part of the lipid bilayer is nonpolar, so that doesn't match. So what water does is it has special channels that are specific protein channels that it goes through. Okay, and they're called aquaporons, are the names of the channels that water is able to go through. And so it kind of acts as like a little bridge that water is able to go across that nonpolar environment. It goes through that protein channel, maybe more, not necessarily a bridge, a tunnel. That's a better analogy. So it's kind of like a tunnel through that lipid bilayer that allows that polar molecule to get across because it can't get across the nonpolar river. So it has to go through the tunnel. And like I said, those are called aquaporons. And the aquaporons are open almost all the time. Okay, they're not regulated. It's not like a little gate that comes down and says, nope, you can't come through. They're just open all the time, and they go through the lipid bilayer through those aquaporons and get into the cell. Okay, so that's how water gets across. There are other substances we'll talk about here in a second. Ions, they do not move readily across the lipid bilayer either, and that should make sense to you because you should know that ions are positively and negatively charged. Okay, so they're going to have some polar components to them, so they aren't going to be able to go through that nonpolar environment. And it's also due to their strong attraction with the hydrophilic side because it's a charge. Those are charged sides, so they don't really want to go across very easily. And so they're going to use channel or transport proteins to get across, to tunnel through to the other side, because they don't have the same polarity. So they got to use a channel as well, just like water did, to get across the membrane. Okay, so that's what I like to think of the membrane as, is kind of like a river. And if things that are similar to what the river's components are, it can swim across the river. But if the river, it doesn't mix with the river very well, then you got to take a tunnel and go across, because it's not the same composition, it's not the same environment. So that's how we get around those issues. Now, the channels that control the ion movement, unlike the water channels, they are not always open. Okay, so it's not free range with the ions. They're regulated. So here's another diagram. This stuff over here just kind of repeats what we've just talked about. Talks about how the membrane is selectively permeable. The phospholipid bilayer allows lipid molecules to go right through. Your smaller molecules, like your water, and this is the part I brought up before, gases such as oxygen and carbon dioxide, they're able to move through the membrane unaided. Okay, so they don't need help per se. They can go through. And when we say unaided, we're not referring to those channels that are constantly open. Okay, that includes them as being unaided. It's like it's not like there's a specific process to get them through. They just go through those channels and boom, you're done. So just make sure you know that if you would have to diagram gases going across the membrane, so like if you had to diagram oxygen going across the membrane or carbon dioxide going across the membrane, you would not use any protein channels to show that. You just show it going right through the membrane. Okay. And then our larger molecules, such as some of our macromolecules, they need help from membrane proteins to help get them through. And so here's something, I really like this diagram. You have your lipid bilayer, you have your proteins, your phospholipid molecules here. So what this is showing you is that non-charged molecules, so this would be non-polar molecules, they can go straight through the membrane. Okay. Water can go through the membrane with those aquaporons. Okay. Oxygen and carbon dioxide, they can go right through. They're not charged, so they can go right through. But charged molecules and ions, nope, they can't go straight through. And then these big macromolecules, look at how big that is. There's no way that can pop through that membrane. Okay, so they can't get through. And so that's why this is kind of a nice little or diagram to keep in your head, because you think, okay, non-charged molecules, those are nonpolar things. They can go through because this environment inside here is nonpolar, so they're the same, so they can go through. The oxygen and the carbon dioxide and water, they can go through. Water just needs the aquaporon, but the channel's always open, so it's not a big deal. These charged molecules and ions, they can't get through because they're polar and they can't get through here, so they can't go through. And the macromolecule, it can't go through either because it's just way too big. Okay, so that's just a nice way to kind of see, like, these are no, these are yes, they can go through. All right, so now we're going to go through membrane transport, modes of membrane transport. Make sure you know table 4-4.1 in your textbook. That's very important you understand and learn the information in that table. So working cells have to control the flow of materials in and out of the cell. It's essential to the survival of the cell. They have to be able to get certain materials in. They have to be able to get certain materials out. And that's the whole function of the cell membrane. That's what it does. It regulates or controls what goes in and out of the cell. And in addition to the plasma membrane itself at regulating this control, there are also those membrane proteins that help with regulating control as well. Your integral proteins that go all the way through that act as tunnels across that plasma membrane to get those molecules that aren't compatible, get them into the cell. Okay, so they also help with that process. So the first concept we're going to talk about is diffusion across membranes. And this is one result of the movement of molecules. Diffusion is the movement of molecules. Make sure you underline, circle, highlight this. Diffusion, movement of molecules. Molecules, molecules, molecules. That's so you don't get it confused with another process coming up. It's always the molecules. It's the particles. We're watching particle movement with diffusion. And so what happens is molecules tend to spread into the available space. And we can see that here in these pictures. So we have water molecules, which is our solvent, and then we have a dye down here, and that's your solute molecules. And when we first put the dye in, it's extremely concentrated down here. And as time goes on, we start to see the diffusion of water and dye molecules. So they start moving around. Okay, it's the dye molecules diffuse through the water solution until we reach equilibrium, and there's an equal distribution of those molecules throughout the whole solution. And so I'm sorry I'm not there to do this demonstration with you, but hopefully you remember in 9th grade when we talked about this, I went in the back corner of the classroom and I sprayed a whole bunch of either vanilla spray smell or sweet pea. I can't remember which one I used with you guys. But I mean, I just kept spraying and spraying and spraying in that corner and sprayed. And then I told you to raise your hand when you smelled the scent. And we watched how that high concentration of aerosol spray, room spray, was in the back corner. And then we watched how it diffused across the classroom. And we saw that those of you kind of in the back smelled it first. And then those of you that kind of sat up in the front and diagonally away from the opposite side of the room, you were the last ones to smell it. And that's because it takes longer for those particles to diffuse across the room. And we could actually see the diffusion of those particles across the room. And then eventually the whole room smelled of the air spray, okay, because it had equally distributed itself within the room. And so it wasn't stronger in one part of the room or the other. It was equally distributed throughout the whole room, and the whole room smelled. So that's kind of the demonstration you want to think about is like if I go spray a whole bunch of room spray in a corner of a room, it's highly concentrated in that part of the room. But as time goes on, those particles will spread and diffuse throughout the room until the whole room is equally, the air spray, room spray is equally distributed amongst the room, and you can smell it equally in all parts of the room. That's diffusion, okay? That is diffusing across within the room, okay? So remember, it's the particle movement we're looking at with diffusion, okay? And diffusion is a passive process. That means it does not require any energy to happen. So no energy is needed. You need to know that. You need to know that diffusion is passive and that no energy is needed, okay? So there's what that's basically saying is ATP is not involved. You don't need any extra energy in order for that to happen. It just naturally happens. So no energy is needed for diffusion to happen. And that's in this lesson, we're going to be talking about all passive methods of transportation. So all the methods we're going to talk about in this session do not require energy. So we're going to talk about another example in the human body of diffusion. So just to kind of review and to continue on, diffusion across the membranes, it's the movement of molecules, you'll hear this term, down a concentration gradient, okay? And you'll hear that term, and that may be a little like, well, what's that mean? So what that means is that we're going from a high concentration of solute to a low concentration of solute. That's what it means to go down the concentration gradient. Normal concentration gradient movement is from a high to a low. That's how normal things flow in the world, okay, of molecular movement. It goes from high to low. So we're going down the concentration gradient. You also need to know that for diffusion, that it goes from a higher concentration to a lower concentration. Okay. Once again, no energy input. We learned that from the previous cell or slide. And so here's another example within the human body. And so it's the movement of oxygen from the lungs to the circulatory system and vice versa. So we can see, here's your trachea where air comes in, then it comes into your bronchi and your bronchioles into your lungs. So here's your bronchioles, and then it comes down into your alveoli, and your capillaries surround your alveoli. So all these little bundles right here are alveoli. These are little air sacs. And then your capillaries encase those alveoli. And so if we look inside an alveoli, you'll see the oxygen that's coming from the air that you breathe. And so we see this high concentration of oxygen here and a low concentration of oxygen here in the capillary. And so what will happen is the oxygen will diffuse into the capillary region because there's a higher concentration here, low here. We know molecules are going to move from a high to a low, and so the oxygen diffuses in. And that's how we can get oxygen into our bloodstream, okay, by the diffusion of those molecules into our capillaries, okay? And that's how that works. So we breathe in, if you breathe in oxygen-rich air, high concentration of oxygen, and blood's flowing past the alveoli is low in oxygen content, the oxygen is going to diffuse down its concentration gradient into the blood capillaries. So it's going, when we say it's diffusing down its concentration gradient, it's diffusing from its high concentration to its lower concentration in the blood capillaries. And that's how the oxygen gets into the bloodstream, through diffusion. Okay, so now we're going to get into selective permeability, and we're going to talk about channel and carrier proteins. So proteins are embedded in the membrane. They allow the passage of molecules into and out of the cell. And so we have channel proteins, two types. We have open channels and gated channels. And so open channels are exactly how it sounds. They allow for the unaided passage of some molecules down the concentration gradient. So they're going to allow these molecules to go from a high concentration to a low concentration. They are always open. They don't need any extra help. They're just there to help the molecule get across the membrane because that molecule probably doesn't agree with the internal composition of the membrane. And so it helps them just go through. So they just go through. And then we have gated channel proteins. And what happens with these is they may require a substance to actually bind with the channel protein in order to open it up. And so examples of gated channels would be your ion channels. So they may need the ions, whatever ions it may be, to actually bind with the channel protein so that it can go through. Kind of like a little, kind of like our swipe cards at school. So it kind of swipes, it recognizes, yep, you're the ion that's supposed to go through this channel. Okay. And it opens up and allows you to go through. Okay. And so this you can see as an example of a channel protein. It's, wide open. We have a higher concentration of molecules on the outside, lower on the inside, so they're going to come right on through. Okay. And what this talks about up here, it says it allows a particular molecular ion to cross the plasma membrane freely. However, in cystic fibrosis, which is an inherited disorder caused by faulty chlorine channels, a thick mucus collects in the airways as well as in pancreatic and liver ducts. And so what happens with cystic fibrosis is these channels, they don't open and close properly. And so these ions don't get across properly, and that causes mucus to get stuck in airways, which makes it extremely difficult for these individuals to breathe. And it can also get stuck in pancreatic and liver ducts, causing digestion issues as well. Okay. So that would be a case in which those channel proteins aren't working properly, and that causes problems. Okay. Here we have carrier protein. So this would be an example of a carrier protein. And it selectively interacts with specific molecules or ions, and that helps them cross the membrane. And you can see here the family of GLUT carriers transfers glucose in and out of various cell types of the body. Different carriers respond differently to blood levels of glucose. And so you can see here, we're still going from a high concentration to a low concentration, but it kind of, like, instead of just being open, like the channel protein, it will like take it in and then help it get across and then spit it out. Okay. It's not like it's constantly open. Okay. It just kind of helps these molecules get across, but it's not like it's always open. Okay. Sometimes it's closed and you can't get across. So maybe you have to be a specific molecule in order to allow it to open up and help you get across the membrane. Okay. So that's we call this facilitated diffusion because it's diffusion by helping things get across the membrane. Okay. And they get across by using channel proteins and carrier proteins as well. And so this slide's going to just kind of reiterate what we just talked about. So facilitated diffusion, it's the use of channel proteins or carrier proteins. The channel protein is always open and allows movement. Carrier proteins will change their shape or their configuration of the protein, and it's specific to the solute being moved through. Okay, so it can change its shape to help that molecule get across. No energy is required for this. Okay, it's a passive process. And we're going down the concentration gradient. So we're going from a high concentration to a low concentration. So in this diagram, this picture down here, you can see we have these little molecules. Most of my students like to call them Shrek heads. So it's a solute of some sort. We'll call it Shrek solute because it looks like little Shrek heads or Frankenstein heads. And so these solutes want to get inside the cell. And so they can't go through the plasma membrane because they aren't compatible. So they got to go through this carrier protein. And so what they do is they go into the carrier protein. The carrier protein molds to the shape of the solute, and then it will push it through. And then you can see right here, it lets it out, and then it closes back up and waits for the next solute to enter. It will change its shape and then spit it out. So it's kind of like it's they're very specific for specific solutes, and they'll mold their shape around that solute so that only that solute can fit in there. So let's say, like, if we had a triangle solute trying to get in this carrier protein, it wouldn't allow it because it wouldn't fit. Okay, so these can be very specific, and that's a good thing because then only solutes that are supposed to go in the cell can fit in there and go in, and things that aren't supposed to be in there and they try to get in there, they can't because they don't fit. The shape's not right. Once again, going into that concept of structure equals function. So if the structure doesn't fit, then it's not going to work. If the structure fits, then it's going to work. Okay, and so that's how these work with facilitated diffusion. So they can become very, very specific, which is good because it can allow good things to go in and not so good things to stay out. It's that selectiveness in action. Okay, so those were the two types of diffusion. We had regular diffusion and facilitated diffusion. So now we're going to talk about osmosis. And this is the movement of water molecules. So it's the passive diffusion. That means it doesn't require energy of water across selectively permeable membrane. That's what makes this different from diffusion, because remember, diffusion was about particle and molecule movement. Osmosis is about the movement of water. Water, water, water. Circle it, put stars around it, highlight it. Make sure you understand the difference between those two concepts. It's very, very important, and that's the key to keeping those straight in your mind. So, with osmosis, if we have the same amount of water molecules on both sides of the membrane, so if we look at this example, this line, this partition right here is a membrane. So we have the same number of water molecules on both sides. Okay, approximately the same on both sides. So that would be an isotonic solution. They have the same concentration. Okay, there's other water molecules hidden back here. We just can't see them all, but it's the same. Okay, now if we would put urea on this side of the membrane, and you can see the urea molecules, it's the big teal color sphere. If we would put that on this side of the membrane, that's a type of solute. So now we have a higher concentration of solute on this side of the membrane, and we don't have any of it over here. So what that means is this side of the membrane has become hypertonic. That means above. Okay, hyper means above. So it's a higher concentration of that solute. And over here, since there isn't any, it's hypo. And so hypo means below or lower. Okay. Hyper is kind of easy to remember because if you're hyper, you're really active and moving about and you have like a high amount of energy. And so that's how I would kind of think of with higher. Like it means I've eaten a lot more sugar, so I'm really excited and moving around and I'm real hyper. Whereas individuals who are hypo are usually really tired. They don't move very fast. And so they're just kind of low in energy. Their energy level is lower. Okay. And so what happens because of this imbalance, remember everything in the world wants to be balanced. They want it to be equal. So in order to cause that to happen, the water is going to flow from the lower solute concentration over here to the higher solute concentration over here. Water wants to go where the solute is. Okay, I always like to say, like, where all the solutes are, that's where the party is, and water wants to be at the party, so it's going to go wherever there's higher solute concentration. It's going to move. Notice that the urea is not moving. Okay, we're not talking about diffusion. That's not what we're focused on. We are only focused on the water molecule movement because we're talking about osmosis. So if you have a higher concentration of solute over here, water's going to move in. And if you get more water on this side of the membrane, what do you notice about the amount? It increases. And so if this is a cell, that means the water, there's going to be more water inside the cell. So the cell is actually going to swell. Okay, it'll get bigger because you have more water volume going inside it and it's going to get bigger. And depending on the amount of solute in here, how hypertonic it is, if you're a cell and you get a whole bunch of water in there, the cell can burst. It can lyse. Okay, and so then you start bursting cells, and that's not a good thing. Now, you could also put more solute particles over on this side to make this become hypertonic, and you could flip it too. Now you notice on this side with the hypotonic side, it's lower. There's not as much water in there. Okay. So basically hypertonic versus hypotonic is comparing solute concentrations. And we're going to get into some, like, this isn't hard, but it's more difficult thought processes, and it's keeping it all straight in your mind. If you can keep it all straight in your mind, you're good. But it may take you a while to get to that point. So you're really going to have to study at home, reread through your notes, read through your text. You're really going to have to work on this because this is not something that you can sit and listen to this one time and grasp it. Now you've got, those of you that took my honors class, you got a little edge because we did talk about it in 9th grade honors class quite a bit. So you've got a little up that, you have been exposed to this. Okay. And now I'm taking it up a notch. Whereas in 9th grade, I kind of introduced it to you and said, here's how it is and this is how it works. Now I'm wanting you to get more mastery of this concept. So water moves from high concentration, water moves from areas of high to low water concentration with the free molecules. Okay. So what that's saying is that if we have that urea in here, the higher level of water on the outside is going to move into where there's lower water in here. Okay, that's what it's saying. The solute concentration, you can see here it's low on this side and high on this side, and the water wants to move towards the higher solute concentration. And so as the solute concentration increases, percent of free water molecules goes down. Okay, and so what that's kind of saying is the more solute concentration you put on this side, the fewer free water molecules there will be, which kind of makes sense because now your solution's got more in it than plain water. And so this right here is pretty key. Water moves from hypotonic to hypertonic solutions. That's real important for you to understand, because what's going to end up happening is I will give you a diagram, or you're going to draw a diagram, and you'll have to label the hypotonic side and the hypertonic side, and then you're going to have to tell me which way is the water going to move, in or out, and why. Okay, so you have to be able to do that. And so, for example, if we looked at this, we would say we would label this side as hypotonic because there aren't any solute particles. We would label this side as hypertonic because there are solute particles, the urea, it's there. And then I would draw an arrow going towards this side of the membrane. And I would say the water would move into, if this was inside the cell, would move into or move towards the right side of the beaker because there is a higher concentration of solute molecules on that side, and water will move towards the higher concentration of solute. And that's exactly how you would do it. That's how you would explain it. Okay. So you would label this as hypotonic. You would label this side as hypertonic, and you would say water would move towards the right side of the beaker because there's a higher concentration of solute on the right-hand side beaker as compared to the left, and water is going to move towards the higher concentration of solute molecules. There you go. And so water molecules surrounding the solute isn't so free. They kind of get jumbled up here. You see that here there's a lot more space, and over here they're real concentrated. So that's what that's trying to talk to you about. But this point right here, water moves from hypo to hypertonic solutions. That's real important that you get that down and put some stars and underline, highlight that. That's important. All right. So why is this regulation of water real important? Well, it's important in many ways. So for our aquatic organisms, they have to actually pump out water because their interior of their cell is hypertonic to water. So the inside of the cell here, remember, it's got solutes and things like that. So the water in its outer environment is going to diffuse into the cell because there's more solute here and the water wants to come in. So there's not as much solute in the aquatic environment that this organism lives in. And so water is constantly coming into its cell. Well, if that keeps happening, the cell's going to burst. And so that's a problem. So what these organisms have done is they have a contractile vacuole in them. And what it does is it'll pump the water back out. Okay, so it acts as like a little pump, like a sub pump, and it will pump out the excess water. And so that's how this organism maintains its water levels and how it stays in homeostasis. Because if it didn't have that contractile vacuole, it would just have so much water diffuse into it, it would eventually just burst and it would kill it. So they have these contractile vacuoles that continually pump out the water so that it can maintain a proper balance of water. Okay. And then plants, as you know, they retain water in their central vacuole. So they have this huge vacuole. And as water diffuses out, that causes the leaves to wilt. Okay. And so you can see wilted leaves, they lack water. Okay. So the water is diffused out. And so when you water them, the water will diffuse back into those vacuoles, and it actually causes a form of pressure called turgor pressure. And then that's what makes the leaves pop back up and look normal again. So we can see nice, healthy leaves here because their water vacuoles are full of water. Okay, and so that's kind of right. That's how they regulate their water, is the amount of water that's in their central vacuoles. And that's how that works. Okay, so now it's question time. So celery stalks that are immersed in fresh water for several hours become stiff and hard. Similar stalks left in salt solution become limp and soft. From this, we can deduce that the inside of the cells of the celery stalks, we're looking for the inside of the celery stalks, inside of the cells, are A, hypotonic to both freshwater and saltwater solution, B, hypertonic to both fresh and the saltwater solution, C, hypotonic to freshwater but hypertonic to salt solution, D, hypertonic to freshwater but hypotonic to the salt solution, or E, isotonic with freshwater but hypotonic to the salt solution. So I'm going to let you guys, we're going to pause the video because you're going to need some time to sit and think about this one. So we're just going to pause the video, and once we'll have the substitute teacher in about a minute, give you guys about a minute to sit and think about it. They're going to, ask you to show your sign. Okay, so you're going to use your sign language. When they ask you, show them your sign if you think it's A, B, C, D, or E. So we're going to pause for you to think about it, and then we'll come back and check your answers. Okay, so you've had some time to think. This is a really tricky question. And the trick is you're thinking of it with regards to the inside of the cells of the celery stalk. And so the correct answer is D. Okay, and so that might have caught a couple of you off guard. Hopefully it didn't. Most of you caught it. So we're going to go through and try to explain why the answer is D. So with the fresh water, when the celery is in fresh water, water is moving in. And the reason for that is because you have a higher concentration of solute inside the celery cells as compared to the fresh water. And so that's going to cause the water to go in. And so you're thinking of this with regards to the celery cells, not the water that's in the outside of the beaker. So let me escape out of here for a second and go to our jam. And I'll try to draw you a picture of this. And I often find drawing pictures of problems that involve hyper and hypotonic extremely helpful. As you guys know, I'm a pretty visual learner when it comes to this stuff. In general, I'm visual. And so if I was approaching this question, there is literally no way I could have answered it without drawing a picture. So I really encourage you to take that to heart. And when you get questions like this on tests or quizzes, sit there and draw a little diagram out and see if it helps you, especially if you're a visual learner. It makes a world of difference. So I'm going to draw my beaker here, maybe. Yeah, here's my beaker. And here's my celery. Okay. And here's my water. I don't want to push too hard on this. So there's my water. Okay. And I know that the water inside here, it's fresh water. So that means it doesn't have any minerals or anything in it. It doesn't have any solutes. Okay. It's like distilled water. There's no stuff in it. So, and then I also know that within celery cells, there's going to be solute, there's going to be salts, there's going to be sugars, there's going to be all those types of things within the celery cells. So I'll use red, and I'm going to try to draw little stars, and those are solute particles. Okay, so these little stars represent solutes. Okay, and so if I look at my diagram here, and I try to identify things that are hyper and hypotonic, the celery, because there's more solute molecules inside of it. Make sure I'm going the right direction here. Yeah. The celery is going to be hypertonic. And I'm just going to write hyper because it's really hard for me to write on this. So it's hypertonic. And if I look at the water on the outside, it's hypotonic. Okay, because there aren't any solute molecules. It's pure water. It's just it's distilled water. There's no particles in it. So if I look at that and I'm like, okay, well, which way is the water going to move? The water is going to move from the hypo to the hyper. So water is going to move into the celery cells. And that's going to cause the celery cells to get bigger. And they're going to be, it'll make the celery more firm. Okay. And so this is in a this is, I'm just going to put DiH2O, which means fresh water. Okay. So in this case, the celery is in distilled water, fresh water. Okay. So that's going to cause, because there's no solute particles outside, or very little, there's some, but not a lot. And there's more on the inside. That makes the celery hypertonic. And so water is going to move into the celery cells, causing them to become more firm. Okay. So that explains this first part, that we can deduce the inside of the cells of the celery stalks are hypertonic to fresh water. Because remember, I just showed you in the other one, when we put the celery in fresh water, there was more solute particles inside the celery cells as compared to the outside environment. So then let's explain why it's hypotonic to the salt solution. Okay, and that's because when we put it into a salt water solution, the water is going to move out. So the cell becomes hypo on the inside and hyper on the outside. So let's take a look at that situation. So I'm going to go to the next slide. So I'm going to redraw my celery here. So here's my beaker, and it's a salt solution. So we're going to put that's so hard to read. I'm so sorry. So that's a salt solution. Okay. So I'm going to draw my celery. So here's my celery. and it's in a salt water solution. So here's my water. And so since I have salt in there, I'm going to draw a whole bunch of salt molecules out here in the water. Okay. Now, it's not to say there isn't salt still in the celery, because it is, because you have those, or solute, not necessarily salt, but solute particles. Okay, so I'll put a few in there. I'll put another one. Okay, but when you look at the overall picture, there's 3 in here and 6 out here. And so what's going to happen if we identify our parts, what's hyper, what's hypo? So the inside of the celery is going to be hypo because it doesn't have as many salt particles, solute particles. And the inside, or the water outside of the celery is going to be hyper. And we know water always moves from hypo to hyper. So in this case, the water is going to move out of the celery cells. And so that's going to cause the celery cells to shrink. And that's when it will become flimsy and soft. Okay. And so in this situation, in a salt solution, the inside of the celery cells are actually hypotonic as compared to the outside. And so that's how we get this. So in a saltwater solution or salt solution, water is going to move out of the celery. So that means that the inside of the celery cell is hypotonic to the salt solution. And so that's why letter D is the correct answer. And the reason students get really confused on this is because in 9th grade, I taught you to think about it from the outside water solution. I never had you think too much about the inside part. And this question, it flipped it on you. Okay. And so now instead of just thinking about the outside, it's making you think about the inside and how those functions work. And that's what I mean with these types of questions. They're going to come at you from all different angles. And if you truly understand this concept, you should be able to think your way through it to figure it out. And that's why when I first gave you this question, I really emphasized the inside cells of the celery stalk, because that's where kids get messed up. They're used to thinking of it from an outside of the cells of the celery stalk. And so for those of you that maybe selected C as your answer, that's how you got C, because you were thinking of it from outside in the solution that the celery is sitting in, as compared to the actual inside of the celery cells themselves. So that's where that was tricky. Okay. And so that's why it's letter D and not C. So I hope that cleared that all up for you guys, because this can be a difficult question. Well, let's try another one. The total solute concentration in a red blood cell is about 2%. Sucrose cannot pass through a red blood cell's plasma membrane, but water and urea can. Osmosis will cause such a cell to shrink the most when the cell is immersed in which of the following solutions? A hypertonic sucrose solution. B, a hypertonic urea solution. C, a hypotonic sucrose solution. Or D, a hypotonic urea solution. So once again, I'm going to let you guys pause the video and think about this for about a minute. Come up with your answer. You might want to do a little doodling diagramming on a piece of paper around you to help you figure this one out. Then the sub will ask you your answer. You'll show it to them using your sign language of A, B, C, or D. And then we'll turn the video back on and I will explain the correct answer to you. So go ahead and pause your video. Okay, you've had some time to think about it. And so the correct answer is A. So those of you that picked A, you're correct. Urea can diffuse freely. So that does not affect the movement of water because it clearly said that urea can pass through the membrane. So it's not, it just goes through, no problem. And so the urea molecule will balance out due to passive diffusion. Okay, it'll just do regular diffusion and even itself out. Okay. However, the question says sucrose cannot move out of a cell. All right, so sucrose can't move. So if sucrose can't move and we want the cell to shrink, we want to find out what will cause the cell to shrink the most. In order to get a cell to shrink, you got to get water out of it. Water will cause it to shrink. And so you have to figure out what kind of a solution can I put that cell in order to get the water to come out, to make it go down, to make it shrink. And so the solute concentration has to be hyper outside of the cell in order to make that happen. Okay, so if we go diagram this out, so we'll draw our beaker. And we've got our red blood cell right there. And we want this guy to shrink. And so in order to get it to shrink, we've got to have more solute particles outside of that red blood cell as compared to inside. And we know if we put all these little particles in here, okay, if this is sucrose, there'll be a sucrose or two molecule inside. They can't move. All right. So in order to get this red blood cell to shrink, you got to have all these sucrose molecules on the outside, so that the outside would be what kind of a solution? Hypertonic, right. So it would be hyper. And the inside of the cell would be hypo, because there aren't as many. And so we know that water is going to move from hypo to hyper. And so water, H2O, is going to move out of the cell. And if water moves out, the cell's going to shrink. And so therefore, we would have to have a hypertonic solution of sucrose in the beaker to make that happen. And so that is why the correct answer was A. And so the water inside the cell would have to move out because the solute concentration has to be hyper on the outside of the cell. And that's the only way you could get that. Because if you take a look at this question B, it says hypertonic urea solution. Well, that's not going to do anything for you because urea moves freely. So it's not affecting water movement because it just moves by itself by normal diffusion. So that's not going to cause the cell to shrink or get bigger. It's just moving by itself. If we would put the cell in a hypotonic sucrose solution, then that would mean you would have less solute particles on the outside of the cell than inside, and then that would actually cause the cell to swell because there'd be more sucrose on the inside of the cell, and that would cause the water to go in. So that wouldn't make it shrink. And then question D is hypotonic urea solution, and we can throw that out because urea can diffuse freely, so it doesn't affect the movement of water. So automatically you can throw out B and D because urea is not doing anything. It's not affecting the movement of water. But sucrose, because it can't get in and out, it does influence the water movement. And so if we want it to shrink, we've got to put that cell in a hypertonic sucrose solution so that the concentration is greater outside, so the water in the cell will come out. And so that's why the correct answer is A. So this is the end of Chapter 4, Part 2, Session 1 notes. If you have questions, please make sure you message me on Schoology. You guys, most of you do a really good job of that, and I truly appreciate it. And I will try my best to get you straightened out the best I can. Like I said, this isn't a hard concept, but it does require thinking on your part. And you have to be really careful not to get impulsive and react by just reading it and then, boom, I find an answer. You can't do that on these questions or you're going to get messed up. You seriously have to take time and think and digest and read every single word and figure out what's going on in the problem, diagram it out before you come to your answer. These are higher level thinking questions and require you to think. That's what makes them tricky. So, like I said, if you have any questions over this material, please make sure you message me on Schoology. Thanks.